Thanks very much. That's a very nice introduction. I think you exaggerated a bit at the end. So this is definitely a first for me, giving a talk at 4:25 on a Friday afternoon, so I'm looking forward to that. I'm delighted to be here. I really want to thank the organisers for inviting me, and my boss, Tony Wood, who I'm standing in for today, also to thank him. It's been an awesome day, three incredibly strong scientists who've done fantastic talks, and now you have me, and I hope you're going to enjoy that. About 3 years ago, James Watson, who probably most of you have heard of, came to visit La Jolla. I'm not one to drop names lightly. In fact, I never do it, but I make an exception this time. When he visited La Jolla, I was the only chemist in the room, and so he was coming down the line, and all the different biologists were introducing themselves to him, and he was going, yeah, hello, hello, hello. And he got to me, and when I was introduced as a chemist, he said, they say that this is the age of biology, but they're wrong. It's definitely the age of chemistry. It's chemistry that is going to be used to solve all the biology problems, which I thought was fantastic for a molecular biologist to me telling me that. I actually think he's wrong, though. I think it's the age of collaboration, and I think we've seen a number of examples of that today. And you'll see through my talk, which is covering drug discovery, a couple of projects, that it's all about collaboration, but that chemistry is at the heart of that. And at the heart of medicinal chemistry is innovative synthesis. Pfizer continues to recruit the absolute very best talented synthetic chemists that it can. It's been doing it for years, and we still do it. Even the last couple of years, we've recruited some great people. And the reason we do that is because the chemistry that we need to do is not trivial. We need people who can really solve difficult problems and make molecules that perhaps a lot of people would look at the molecule and run away or laugh. I hope I don't need to explain to many people the importance of synthesis, but we'll see. You can ask me at the end if you need to explain. I'll talk about two projects. The first project is where we're trying to come up with a cure or a treatment for Alzheimer's disease. The second one is a treatment for non-small cell lung cancer. The first project is a project run out of the MedChem group over on the East Coast. Now, I've been a bit of a voyeur on this project, so I've followed it, but I am talking about someone else's work. But it's a great story, and so I'm glad you've stayed here this late, because it's going to be good. The second story, I can use the term "we" throughout that talk, because this is from a La Jolla project. I kind of gave the punchline at the beginning that it's an essential enabler for today's challenging drug targets. We need to be able to make difficult molecules. Both for a drug lead discovery and the optimization process, we really need to rapidly be able to cover even difficult to access chemical space, because we're trying to solve so many different problems, molecular problems in parallel, that we can't just use simple molecules 99% of the time. And then when we need to, when we're successful and we have a candidate to get to the clinic, particularly in oncology in recent years, if you start to see really exciting activity early in the clinic, what happens is there's a huge call for acceleration. With FDA, if it's breakthrough activity, we'll want to move that compound as quickly as possible to approval, as long as it's safe and given the breakthrough efficacy. And that puts incredible stress on the sort of traditional compound supply chain in terms of just how quickly you need to be able to make kilos to hundreds of kilos of compounds. So we need to be thinking about that way earlier than ever before in medicinal chemistry. And that means we need robust and efficient routes for scale-up because of that accelerated development. Let's start with the Base 1 project. And can everybody hear me at the back? Because I'm going to kind of move in and out away from the microphone, but it's all good? Okay, thanks, Ben. Inhibition of Base 1 is really a compelling target for medicinal chemists to work on in terms of Alzheimer's disease. I'm not here to explain to you the whys of that or the details of the biology, but there's a very strong rationale for inhibitors of Base 1 based on human genetics. Base 1 is the beta-site amyloid precursor protein cleaving enzyme, which cleaves the full-length amyloid protein and ultimately liberating, after a gamma secretase, A-beta, which is strongly implicated in Alzheimer's disease in terms of deposition and formation of the amyloid plaques. BASE1 is a really challenging medicinal chemistry target. It's in the brain. We need to inhibit this enzyme, so CNS penetration is required. You're going to be treating elderly patients. You're going to be treating for a long time, chronic dosing. So you really need to have really good safety, and you need to avoid drug-drug interactions, because these are probably people who are going to be on many other medicines. You need to hit, and partly because of the safety, you need to have selectivity versus other Esparto proteases, especially cathepsin D, which is a very similar protease to BASE1, and linked with significant eye tox if you inhibit cathepsin D. And then, again, because of the safety, we need to be hitting really very selective compounds that don't hit other off targets. And some of the problems that came up in our project were inhibition of hERG channels, which can lead to cardiac problems, and muscarinic antagonists. So we're after an inhibitor that's suitable for long-term dosing, and we need potency, selectivity, we need to get into the CNS, and we need to have reduced safety liabilities. And these are the key challenges. Very simply, they need to get to work in the brain. To do that, you have to get past the blood-brain barrier. Blood-brain barrier restricts access of drugs to the central nervous system. Fortunately, as medicinal chemists, we know how to design inhibitors to beat the blood-brain barrier, but it means you have to be really disciplined in your design. You need to keep the size, set the molecular weight down of the molecules. You need to target an optimal lipophilicity. It's kind of a bit of a joke, this, but log D, a lipophilicity of around 2.2 usually is the optimal one. We see that time and time again. But usually generally somewhere in between, say, 1.5 and 2.5. We need to minimize the number of hydrogen bond donors, because these increase the chances that your compound is going to be a substrate for a transporter in the blood-brain barrier, and also slow down the passive permeability through the membrane. And we need to avoid strongly basic groups, because they're really linked with a high chance of being picked up by a transporter and excluded from the brain. So we're talking about Esparto Approaches, and when you look at all the early projects that tried to get Esparto Approaches inhibitors like renin, these are all great big hairy molecules, look more like peptides than small molecule drugs, lots of hydrogen bond donors, very polar. Really, you just wouldn't expect one of those to get into the brain. So really, it took a long time, say a decade of research across the industry before they came this thioamidine. Now I've got three things, and so I've got a problem. I'm going to get rid of the water here. You know, thinking, talking, and operating two instruments at the same time, any time of the day is difficult, but it's really hard now. So this thioamidine here emerges as a preferred core binding motif. This on the right here is a picture of the structure of this compound bound into in green bound into BASE1, and in yellow it's actually bound into, it's modeled into a cathepsin D structure, and you can see that there are differences between several residues, and thioamidine binds to the two catalytic aspartates. The strategy that the team took, because of this focus to really keep molecular weight down, was to really focus on efficiency, so particularly atom efficiency. So make sure that every atom that you're going to put in to your compound is really going to be delivering something of value in terms of potency or selectivity. And so they truncated to a minimal binder. So other compounds that were around from screens and in the literature had substituents here, substituents all over, were much bigger molecules, and so they stripped away all of the substituents to get to this minimal fragment, and then the idea was to build efficient and selective potency, particularly focusing on cathepsin D. This compound, people might look at that and say, well, so IC50 is 36 micromolar, not a very exciting start point, but it is very small, and the ligand efficiency, I just don't have time to explain that, but For those of you who don't know, it's a metric we use to describe how efficient the atoms are delivering potency. And 0.4 is kind of reasonable. And it was selective over Cat-D. And this is a cell-free assay, that data. So there were actually three ways that the team thought they could potentially build in potency in a way that would give them selectivity. There's actually three key areas of the protein where the cathepsin D is different to base 1. The first one would be to disrupt an arginine asp salt bridge that's present in cathepsin D here. Second one would be to engage the phenylalanine backbone carbonyl, which is only present in base. And then the third one would be to, and this was a little more subtle, would be to interact with the flat region, the S2 region in base 1. And the small difference here were that that region in base 1 the protein was quite flexible, whereas there's more structure in the Cat-D region there, and so it was expected that you maybe would be less tolerant to putting substituents or testing that vector. And really just for reasons, I think that of efficiency, to reach this one, you're going to have to use more atoms. So strategies in this is a vector, if you like, the direction, would use more atoms, 10 angstroms away, 8 angstroms away. The closest opportunity, or the strategy that was most aligned with maybe efficiency, making smaller changes, getting higher atom efficiency, was this number 3, and that's really where the project went, and you'll see that was ultimately successful. So when we're doing structure-based drug design, we're looking for vectors, and vectors are kind of, if you like this, like it might be a CH. So it's somewhere where potentially if you can do the synthesis to modify that molecule, you're going to be able to grow the molecule in that along that vector, and then reach to somewhere that you want to interact with. It could be a lipophilic pocket, it could be form a hydrogen bond, it could be to push against a protein you don't want to inhibit. And that's a common thing to do in structure-based drug design. So the very first thing that they tried was, it's a lovely example of efficiency in action. Literally, what am I doing? Is that your most least favorite term? It's mine. But I have two 17-year-old daughters, so there you go. They added only an oxygen, 2 carbons, and 2 hydrogens, so just 3 heavy atoms. They formed a new ring here. This is it, the new ring. And this reinforced the bioactive conformation and starts to push up towards the flat vector. So they went from 36 micromolar to 5 micromolar, and this compound hadn't shown any activity in a cell assay, whole cell assay, and this compound now showed activity 265 nanomolar. Throughout this series, it turned out that the compounds were more potent in the cell than they were against the enzyme, and that was a good correlation all the way through. And the selectivity was still very high. And the ligand efficiency was pretty much the same. Now, what you often find is as you grow molecules, the ligand efficiency will go down, can go down quite a lot. So you're doing actually quite well as you grow a molecule just to maintain that ligand efficiency. And what they noticed was that in this structure, then the green compound is this compound here, and this tyrosine 71 is kind of sitting across here. But in the structure for the bicyclic molecule here, this tyrosine is flipped back out of the way, and this has kind of generated a new pocket here. And so what they did next was do another really efficient thing, which was to essentially put a methyl, although in this case it was a methyl with a fluorine in there. I'm sure they did make the methyl as well. And so really they added only CHF, and they got a 5x potency gain. And again, what they were doing was probing the vector further up into this pocket. And this compound then, again, so you've got a little bit more molecular weight. Ligand efficiency has stayed about the same. It's better if you can increase it, but it's rare to do that. because you'd have to get everything continuing to be perfect in terms of interaction with the protein and strain to increase ligand efficiency. And this compound now is kind of quite respectable, 61 nanomolar in the cell assay and very good selectivity. So some of you are probably thinking, well, this is a pretty complicated structure. More chiral centers than you might have expected in a drug talk. So what was the synthetic strategy to access this kind of late-stage intermediate? In a retrosynthetic sense, the first idea was to control the cis ring fusion by a stereoselective aryl metal addition from the convex phase of this bicycle here to give you the aryl group axially and cis. And it was anticipated that this system could come in a stereo-controlled sense via a 3+2 intramolecular cycloaddition, where the R1 group would be in the equatorial position in a transition state and give you the 1,3 stereo control that you require. And it was anticipated that you'd be able to build the nitrile oxide from an R homolytic alcohol, which you had made by accessing the chiral pool to get the R enantiomer via an epoxide and a Grignard addition from R- glycidyl benzoyl ether. And then finally, in the final steps, you would cleave the NO bond and react with isothiocyanate to give the thioamidine. And in particular, they were interested in testing this vector further by putting small heterocyclic groups in here, which would not increase the molecular weight very much and would particularly keep the lipophilicity down in the area where it was needed to be. And I think pretty straightforward heterocyclic chemistry via the acid and the aldehyde through to oxazoles, isoxazoles, and the pyrazole as well. And mostly using small groups, so you'll see a lot of methyls. In the world of efficient medicinal chemistry, the methyl group, or the methyl group you may prefer, is your friend. So these heteroaryl substitutions did improve potency and maintain good ADME. What they found is they did some in vivo testing with the CHF compound, the precursor lead, and I think they had to dose very high in the rat to get exposure. And I suspect that there was, because of off-target activity, they actually got convulsions in the rat. And so this compound really, they weren't able to test the hypothesis of inhibiting base 1 in this model. And so the objectives at this point were to improve the potency greater than 10-fold, but do that in a way which didn't negatively impact clearance and kept good brain penetration. I guess the best thing you could say from the safety side is at least they were getting into the brain, because you don't get convulsions usually unless you're in the brain. So the design strategy was a simple one, which is really to optimally fill this induced pocket. And here's an example with the pyrazole. And so these look fantastic. You narrow down in terms of the whole cell assay, 3 or 4 nanomolar in particular. The methyl substituents really boosted both potency, efficiency, ligand efficiency, and LIPI, so atom and lipophilic efficiency. LIPI jumping up to 5.7, 5.5, 3.9 ligand efficiency, even though you're increased, you've 0.39 ligand efficiency, even though you've increased the molecular weight somewhat. And the problem here is indicated on anything that's red. And basically, these two compounds were very, very unstable metabolically, and they were all of them were substrates for CYP2D6. And the issue with CYP2D6 is a high polymorphism in humans, and so you can get very variable PK if you have a drug which is cleared by CYP2D6. Some of these compounds are also potent inhibitors of CYP2D6, and so this can lead to drug-drug interactions where if the patient's being dosed with another drug which is cleared by CYP2D6, you'll get higher levels of the drug, and that could give you safety problems. And actually, if you have, you could also have a drug-drug interaction, but if you were being co-dosed if your drug was being co-dosed with another drug which inhibited CYP2D6, again, you could find out that your drug was going to be much higher levels than you expected in certain patients or in those patients that are treated with the other medicine, and that would be a problem. So, what would the design strategy to solve the 2D6 issue be? This is pretty challenging because all of these heterocycles are subject to the problem, and some are cleared rapidly, particularly with the methyls. And so many people would think, well, we need to try different heterocycles. They maybe start to try and make bigger molecules and forget about the efficiency and come into problems. Maybe think about putting a fluorine or some other way of blocking the soft spot. For these two, it's likely that this methyl group is being metabolized. In this case, this is being metabolized to the NH. However, there's a much more subtle, successful result that they came up with. It was another methyl. Actually, now you're looking about getting a really complicated molecule with, what is it, 4 stereogenic centers now. A very efficient way to solve the CYP2D6 liability was just to add a methyl group here. So you had just CH2 because obviously you're losing a hydrogen there. And so if you compare where R equals hydrogen, potent, good selectivity, but unstable and strongly metabolized by 2D6. Just going to the methyl, maintains potency, selectivity, now stable, and a much more balanced cytochrome P450 metabolizing enzyme profile. These are the enzymes that metabolize compounds when you dose them to humans. And lipophilic efficiency comes down a little, ligand efficiency comes down. What you've done here, though, is you've solved what looked to be a really critical problem. So now you can come back and do further optimization to bring the lipid and the ligand efficiency, maybe a little more potency, depending on what you see in vivo. So this was easy enough to design, right? Because in fact, it's the first lesson that I give to people. If there's somewhere on your molecule you can put a methyl in, put it there, because it's always efficient. You never add a lot of molecular weight. It can often have a big effect on the conformation of molecules. They can often just pick up a small binding pocket. So it's a really high probability tactic to use. And I don't know whether they did it, but there's at least 2 more places where they should have put a methyl on if they haven't yet. Actually 3. 3 there and actually another 3 there. But remember, you've got to keep the overall molecular weight and lipidity down. So don't go crazy with the methyls. So the question is, does the chemistry, how does the chemistry work, and can they use the previous route to actually control that stereocenter as well? And the answer is obviously yes, otherwise I wouldn't be talking about this project. So here's the first route is a flexible route to a key intermediate. Pretty standard conversions here till you get to this step here, where you then treat the hydroxylamine imine with the very high-tech bleach and dichloromethane. I don't think that's a green mix there, but in future I'm sure that could be fixed. And we get very high stereoselectivity through a transition state which keeps both groups in an equatorial position. You have got the wrong stereochemistry there, though. The opposite to what you want. The next step was converting in this case, the OH to fluoro, and that was using actually some flow chemistry using either Exel, Fluori, or DAST. Variable, but potentially pretty good yields depending on the scale, I suspect. And you could make them, and there was also a way to access the methyl this way, but a quicker, shorter route to the methyl, R2 equals methyl, just to start with this compound here where you have the methyl right from the start. Pretty good yields, same stereochemical outcome. And the next step is to do add the aryl lithium reagent, potentially much higher yield for the CH2F than the methyl in this case. I suspect more optimization could have been done to increase that, but again, you get the very high stereoselectivity to get the all of your stereocenters are in place now. And so what you need to do then is, this one needs to invert, this sensor needs to be inverted. And of course, you're going to do an SN2 reaction to make the thioamidine, so it's perfect. It's a good job that they got the wrong isomer in the first one, and it was the one they expected. And so the final steps then are reaction with the benzoyl isothiocyanate, and then Mitsunobu conditions to close and do the inversion, SN2 inversion, triphenylphosphine dead. I just think that's really nice chemistry. Now, why did that methyl, which was miles away from the place in the molecule that was being metabolized, the heterocycle, why did that work? Most likely, this is explained by the structure here. If you look at the structure, actually it was generated by Scripps with, so this is the 2D6 protein, here's the molecule with the CH2 there. It's this vector coming from the CH2, where we put the methyl is really close to this helix, and so there's a high likelihood that the kind of remote effect of the methyl group this compound really doesn't like to bind to 2D6 anymore because you get the clash with the methyl group. And we have seen this in other cases as well, where you don't have to necessarily get rid of a soft spot if you can kick the compound out of the metabolizing enzyme by fixing another part of the molecule. This new vector also provided a means to optimize hERG selectivity. The therapeutic index for hERG was 49 times, and because we're going to go into old people for a long time, we're going to need better than that. And what this basically just shows you the analysis of all the Pfizer hERG data set. So these are the most basic compounds, pKa greater than 8.5. These are the least basic. These are the most lipophilic. These are the least lipophilic. You can increase your chances of success of getting a compound which doesn't have strong hERG activity by working in this sort of region, 6.5 to 7.5. You can see you've got a much higher chance of success. And working within this lipophilicity space. Compare that to working down here, for example, where you could get a compound, but the chances are you're going to find that very difficult. And also for a subset of all base compounds, they showed that between 7.5 and 8.5 compared to 6.5 to 7.5 for the basicity of this group, you had a much better chance. And so that's why that previous route, they wanted to make the fluoromethyl as well as the methyl. And this reduces the pKa from 7.8 to 7. And this is a very commonly used and often successful tactic in medicinal chemistry, which is to introduce fluorine to modulate the basicity of other parts of the molecule. And here now the potency was increased, and the therapeutic index now is 370, which is much better. So I'm pretty sure this is the last slide on this for this part of the talk. And basically these fluoromethyl substituted thioamidines, they balanced based on potency, selectivity, and all the property alignment that was required to get really good brain penetration and low clearance. So good log Ds, they go 2.2, 2.4, 2.2. And I put 2.2 on before I noticed that they actually are that way. It's uncanny. It happens time and time again. People who work at Pfizer know that. They've seen it. You guys are probably thinking I'm crazy. And some of you are probably thinking, what is log D? Anyway, well, you can come up and ask me about that afterwards if you like. So focusing on designing for efficient potency efficiency using structure-based drug design and fantastic synthesis is what enabled them to get compounds that they were able to dose in chronic A-beta lowering models in vivo, and they got robust central efficacy following acute transmission administration. This was a big and is a big project for Pfizer, so the chemistry team, enormous. Congratulations to all these folks, all working on the East Coast. A lot of structural biologists, computational chemists. It's a collaboration. We have CRD and farm side, biology, safety, pharmacodynamics people, and external collaborators as well. Now for something completely different. We're trying to cure Alzheimer's. Let's see what we can do with cancer as well. So Xalcori is a compound that came from the Pfizer La Jolla labs. It's an inhibitor of anaplastic lymphoma kinase, and it was actually approved in 2011 as really the first new drug approved for lung cancer in the US in more than 6 years. Breakthrough achieved, breakthrough efficacy, rapid approval, and our pharmaceutical really had to run faster than they've ever run before to make enough drug and complete the package needed for registration. But this is not about that story. That's just to convince you that inhibiting ALK is really useful. It's a really good cancer target, and crizotinib works. However, like a lot of cancer therapies, patients, although they have most patients have a remarkable response, and the median improves survival is just under a year, and so that means a lot of people go on 5 or 6 or longer years from a situation where before, without this drug, they would be dead, and they're still alive. But most people will be starting to fail. Their cancer will be resistant to crizotinib within a year. Now, there are 2 alternative paths to disease progression. One is that the cancer contains ALK enzymes that have mutations, and there are multiple of these, multiple mutations, which mean these enzymes are not inhibited well by crizotinib anymore. In fact, the main reason is they're just much more catalytically efficient and a higher turnover enzyme, and also some of the patients, they have a much more highly expressed levels of EML4 ALK, which is a fusion protein for ALK, which is really the oncogenic driver in this disease. It's a very, very nasty lung cancer. A lot of metastases, particularly into the brain, and so some people fail because they get brain metastases, and crizotinib was not designed to get into the blood-brain barrier, and so really the brain is a safe harbour for metastatic tumour cells. And so if we're going to help these people, because we want to, who are failing on crizotinib now, we need to target resistance. We need to come up with a compound that hits these resistant mutants and a compound that gets into the CNS. This is a typical sort of profile that we go for at the start. We want an oral, once or twice a day dosing, really good potency against both the initial EML4 ALK and all the crizotinib resistant mutants, get into the brain, and have very high selectivity, because we're going to be exposing patients' brains to the drug. And if you have off-target activity, that could really reduce your chances of really bringing a medicine that's going to make a difference to people. So we have cell-based assays for measuring how effectively our compounds are hitting the mutant. This is the gatekeeper mutant cell-based assay, and we're looking for less than 50 nanomolar in that assay compared to 800 for crizotinib. As you'll see, we actually end up doing quite a lot better than that in the end. And we're looking for a compound that has low clearance. And these two assays predictive of whether we're going to get into the brain or not, permeability and efflux. We want low efflux. We don't want our compounds to be a substrate for the efflux transporters, particularly the MDR or BRCP, which are going to kick our drug out of the brain. So this is what we're looking for. The challenge is that we need to improve all of these properties and avoid any new issues. And so, as you'll see, we actually took the exact same approach as the previous project. And in fact, we were probably working on the projects around the same time. So just a really ruthless approach where efficiency is at the top of the chart in terms of strategies. So we did overcome all these challenges, and we did discover lorlatinib, which is this molecule here. It's in the clinic in phase 2. This was crizotinib. How do we do this? This is just the overview. We solved the crystal structures and created assays for all of the resistant ALK mutants. Then we attempted to optimize several non-basic or only weakly basic acyclic series derived from crizotinib. One of the main reasons crizotinib has a high MDR efflux and doesn't get into the brain is because it has a strongly basic group here. I've already told you earlier on about how to get compounds into the brain. We realized it would not be possible to hit our target profile ultimately without a really innovative step jump to the new lead series. We made a lot of compounds here and worked hard for quite a while, and we were not getting close to where we needed to. So the innovative step jump came where, using structure-based drug design, we designed and synthesized macrocyclic series and then used that efficiency optimization and structure-based drug design to hit everything we needed to hit all in one molecule, and that's lorlatinib. In doing that, we designed and synthesized 1,600 new compounds, 18 chemists, and in 30 months from the start of the project to delivering the clinical candidate. Where did the idea for the macrocycle come from? A couple of different compounds, co-crystal structures in wild-type ALK showed that given the conformation that these acyclic compounds made, the two ends of the molecule came close together, and we could envisage putting efficiently just a few atoms, right? That's what we're trying to do to make a ring here. And this would give us a novel template that we expected to be give us a chance to really optimize potency. And we found very early on that some of the first examples we made really took us into a different league of potency compared to the acyclic series. It also would help us to improve blood-brain barrier permeability, where we would be masking some permeability, masking some polarity, getting rid of some potentially some donors, having fewer radical bonds and a lower surface area, more globular molecule. When you're trying to make this a 12-membered ring, if you're trying to make rings down to 7 or 9, it's a real hard job. It starts to get a little bit easier, still tough at 11. 12, it's not as easy as making a 5-membered ring, but it is doable. Synthesis. We wanted a conversion and modular synthetic strategy so that we could enable optimization and hopefully scale up with the same route. This is a gross simplification. You saw a lot of chemists working on that project. There were a lot of things being done in parallel, a lot of different series being done in parallel in terms of the macrocycles. I'm just really selecting the path which ultimately led to lorlatinib. So this is a selection. So for example, the design team will be designing a molecule like this. They're interested in testing vectors in this group, here, here, and here, so starting with methyl, of course. And here would be CH or nitrogen. So retrosynthetically, you can see breaking these two bonds to give you a kind of modular approach where you have two modular components. And you can, in terms of putting these back together, you can either envisage closing the final bond, first doing a metal-metal coupling here, and then closing the final bond with a macrocyclization amide formation, or you could do it the other way around. And so we needed to figure out what was going to be the best way to do that. This looks like really simple chemistry. I'm pretty sure that there was a lot of work that went into this and a lot of optimization. All I know is I didn't do any of it, but I'm very, very grateful to all the chemists in the group that did. And there's a very, very good route to access two key module 1, as I call them, pyrazole intermediates with a bromine and a hydrogen there, which would give us flexibility when we came to test the aryl-aryl coupling chemistry. And this was scaled to 20 kilograms, so this was pretty robust and good chemistry. And the module 2 starts with which is this one down here, starts with an asymmetric reduction of the benzophenone here. In fact, good yields and excellent E, and in fact, for the large scale synthesis of this intermediate now as an enzyme, keto reductase reaction is used. And then making the mesylate, displacing with the hydroxyaminopyridine. Hopefully they've got the yield higher than that by now. I imagine they would. If not, someone needs to be working on it. Then the and this is really nice, carbon methoxylation with palladium, good yield, and then bromination gives you the second piece that you need. Now, I don't know how much reaction screening that you folks have seen, but we have a very large group at La Jolla that does it. Actually, it's just one person, but the technology, actually two now. The technology is so powerful. I call it rapid reaction screening. What this is showing you is the yield as determined by product yield determined by mass ion count and the product yield by UV area. And each one of these dots is a different reaction. And what you're looking for is something up in the top right-hand corner, because that's the best set of conditions. So using, and the chemistry that's being tested is, there's 3 aryl bromides here. Aryl bromide plus the boronate reagent to form, coupling to form the pinacol reagent to form the aryl boronate. Using potassium acetate as base, screening 6 catalysts, 6 different catalysts, and 4 solvents, 3 here. That makes 72 reactions done overnight, and all this is done in parallel. This is something that used to take me about 6 months, I think, when I was doing my PhD or postdoc. Now it happens it seems almost instantaneously at times. It's incredible. And from this rapid reaction screen, it was determined that this one never formed any of the desired boronate. This one was pretty good, but really the highest yield was the bisphos bromo pyridine. And potassium A as the preferred catalyst system is actually a palladium acetate plus this very hindered phosphine. And then another rapid screen was done, because a lot of these reactions, you try them the first time and you get really low yields, and I think you could be put off, and then you could try another 10 different conditions and you'd still be put off. But if you try 96 conditions very quickly, you cover space so quickly that you get a really good idea, is this a possible reaction? Because if you get nothing in 96, usually we'll walk away from that. We'll have covered all space. And again, that can be done very quickly. So in this case, 8 catalysts, 3 bases, 4 solvents, 96 reactions. And the best conditions were, as here, toluene, cesium fluoride, sodium hydroxide, and this palladium. And the key thing here was that the optimal catalyst was actually different to the potassium one. Originally, we hoped we'd be able to do both formation of the boronate and the coupling in one pot. And whenever that was tried, they got very low yields, and really it was that we couldn't find a palladium catalyst system that worked equally well for both. And so this was what the screening produced, which was very, very powerful. So using that knowledge, the fit-for-purpose synthesis of lorlatinib went from this compound, and this was done just to show it's a pretty serious scale. Protection, form the boronate, do the palladium coupling with the second screen results to form the compound here, ready to hydrolyze, deprotect, and hydrolyze, deprotect, and then cyclize. And pretty good yields. And then for this last one, 49% for the 3 steps. It looks so easy when I put it on a piece of paper like that, but you really need very good chemists working very hard and with real talent to be able to make chemistry like this work. And it has to be done quickly. And this was the route that was used by a process group to deliver material right the way through first in patients. I think the only modification was they used potassium trimethyl silanate instead of KOH, so got a cleaner ester hydrolysis. For the case where we had nitrogen here, so the pyrazines, we tried the coupling using bromine or bromine here, so a palladium coupling, or even what was with hydrogen there, kind of almost a CH activation coupling, again using palladium. 36%. 9%. So in this case, I think for when these animals were made, it was this route that was used. But ultimately, these didn't yield clinical candidate quality molecules, but just showed that the chemistry was flexible enough to access both types of macrocycle. And in fact, with a little more optimization, and again using that rapid reaction screen mode. A pretty good overall yield, including 75% for 69% for the really novel and funky coupling from the pyrazole hydrogens through to the bromo. 24% over 4 steps and chromatography at each step, and they made 40 grams of this compound as well. So we're not just making milligrams in the lab; we need to make serious quantities of molecules for testing in animals. So in fact, the next generation synthesis of lorlatinib, I showed you one earlier on, which was the first one that was made for the early studies. With more optimization, this route's shorter, gets rid of some problematic reagents, it's greener, and they've actually made up to 3.2 kilograms here. And so this one starts with this piece module, pyridine, reacting, coupling to form the amide with a dehydrating reagent, good yield, and then doing the CH, we call it the CH activation chemistry across here, good yield, 67%, and then just forming the HCl salt as the last step after you take off the two bot groups, 39% overall yield. So for molecules which are so complex, chiral centers, 12-member ring, it's an amazing achievement to make these. I think a lot of medicinal chemists, if you drew that structure and said, we're going to make a project where you're going to make compounds that look like this, they would be a little bit nervous, I think. And that's where the innovative synthesis comes in. Very quickly, because this is more about the synthesis and the design, we did understand very well and use an understanding of how these molecules were fitting into the ALK active site. We had a lot of crystal structures to really understand what was going on and really optimize in the most efficient way the molecules. So you have a methyl here. This fills a small lipophilic pocket here. This carbonyl interacts with a structural water. This fluorine fills a small lipophilic pocket down here. And almost as importantly, I believe, it polarizes the two adjacent hydrogens. And so these are both pretty close to forming hydrogen bonds and non-standard hydrogen bonds with a couple of carbonyl groups there. And there's just a lot more of this molecule is buried in the actual binding site compared to a lot of other kinase inhibitors where you'll have great big groups with piperidines and amines and things hanging off out into the solvent. This is a very efficient molecule. There's the standard donor acceptor hinge interaction at the back here. This methoxy group was filling a lipophilic pocket here. And the electron donation from the methoxy group also enriched this pyrazole acceptor, which was actually interacting with a pi, a CH, again, almost a non-standard interaction of a CH with a really electron-rich pyrazole. And the methyl was interacting with the G loop. This compound here, it's a 0.2 nanomolar Ki, 2.1 nanomolar in the cell. You remember I was talking about trying to get something better than 50. A little bit more lipophilic than ideal, but a great compound. Very high lipid. And I haven't talked about FQ. This is if you're really following ligand efficiency closely, you would use FQ. And if you're interested in knowing what that fit quality, it's a better way for measuring ligand efficiency if you're really varying the molecular weight of your series significantly. And there's a good paper to read on that. Very quickly, one of the things we needed to do was be selective, and particularly against the TRUC enzymes, which are very close in structure to ALK, with a big difference being that the TRUC-B has a tyrosine here, where ALK has a smaller leucine. And so this was a vector. This is a selectivity vector. We could grow here. We would push against any of the kinases which have larger substituents here, like a Phe or a tyrosine. And over 60% of kinases have a large residue here. So that was an immediate way of providing broad spectrum selectivity, and that did pan out very well. In terms of the fit quality, so this is the atom efficiency, and this is the lipophilic efficiency. So high is good, more efficient is good. Gives you a much higher chance of achieving everything in one molecule: permeability, clearance, and brain penetration. And each one of these is a compound. The acyclic series we started with, the grey, and the macrocycles are green. You can see that you've really pushed up to this is the box where we need to be to get drug-like clinical candidate quality molecules. We only just got to the edge of this with some of these molecules, and nowhere near a clinical candidate, the acyclics. But the macrocycles quickly peppered this area. In terms of a lipid plot, so this is the lipophilicity of the molecules, higher lipophilicity here, lower. This is the cell-based log IC50, so these are more potent. This is 1 nanomolar. These are compounds that are more potent in cell than 1 nanomolar. And this is the lipid line. And so these are the most efficient in terms of lipid. You can see again that the macrocycles compared to the acyclics are really ringing the bell there. And then this then is the lorlatinib. What do we achieve compared to crizotinib in terms of hitting the wild-type ALK? we're 62 times more potent, 1.3 nanomolar. In the cell, we're 34 times more potent against the resistant mutant, 21 nanomolar compared to 841. In ROS, we are 300 times more potent. ROS is an enzyme very similar to ALK, which is also forms a fusion protein and is responsible for a significant number of patients' lung cancer. We had really good selectivity against TRUC. We weren't active against c-Met, which was the original target for this compound. And the indicators for brain permeability were really right where we needed them to be. And in rodents, we got good brain to free plasma ratios. Bit closer to 2.2 than 2.4. I'll take it. I mean, this is a marvelous compound in terms of the safety profile in animals. This compound was awesome. And so far, touch wood for the patients, it's working very well in humans. And here's an example. This is a CAT scan of a brain of a 40-year-old woman who was diagnosed with ALK-positive non-small cell lung cancer in December 2012. Was given crizotinib from January 2013 to December 2013, almost a year. And then alectinib, which is a more potent ALK inhibitor than crizotinib. But alectinib does not hit all of the mutants. What I haven't shown you is data that lorlatinib hits all of those resistant mutants. It hits mutants that no other compound that we know of hits, and so it really is going to have a broader efficacy against those resistant, against patients whose cancer has become resistant to even the second-generation agents like alectinib. And so alectinib was given from January to November. And then in December, lorlatinib was started, 100 milligrams once a day. And so I don't know what's happened since July 2015. This is when this was shown. You can see here, these are the brain tumors, and they've completely disappeared. And this is what you see with ALK inhibitors. Normally you're looking at lung, now we're looking at brain. So it's a real proof of principle that we have a compound that's safe, gets into the brain and hits. We don't know, of course, whether these are what the mutants' status of these are, but as the clinical trials progress, we'll find out more. And in fact, I can see here that I hadn't spotted this. I just added this slide yesterday. There's the G1202R mutation, which is the one that our compound is the only ALK inhibitor that hits this mutant, was detected by circulating free DNA analysis prior to the start of lorlatinib. Another big team, an excellent team, fantastic collaboration. There's a lot of science going on in there, structural biology, pharmacodynamics, enzymes, protein chemistry up the wazoo, making all those different mutants. So you have assays against all the resistant mutants, you have crystal structures, and if you're going to try and come up with a miracle drug for people, because if you tell someone, if someone knows they're going to die, the doctor tells them they're going to die, and you're able to give them a pill that they just take once a day, and then if they get another year of life and quality of life, we know by personal experience, because they tell us, that is something that they're so grateful for. And can you go back and get me something that will give me 2 years? So we have to keep trying. And that's my final slide, I think. So thank you.